Good evening, everyone, to the listeners in YouTube as well as in Facebook. Today, we are starting with our vertical three, the practical sessions focusing on open and proprietary softwares related to drug discovery. And today, we will be focusing more into molecular property diagnostic suite that is called MPDS and open source tools for computer aided drug discovery. I would like to invite Professor Narahari Shastri, who is a director of CSIR-NEIST, Jorhat, to please proceed with the session. Over to you, sir.
Thank you very much and good evening all of you. Today we will be witnessing an effort that is made in my group for the last decade or so in developing an indigenous software. But however, a lot has to be done, and I would like to describe that any tool development or instrument development can be perceived in different ways. We have vertical 1, vertical 2, vertical 3, and if it is developing a aeroplane, vertical 3 will be like the mechanical design and prototype making, and maybe vertical 2 is the engineering aspects and the principles and the design aspects and what are the techniques that are required to make an aeroplane. And probably vertical 1 is like principles of automobile engineering and physics and other mathematical principles. Why I say like this is, if you look at any product, like for example a motor car. If the motor car has a problem, where do you go? You go to a mechanic and the mechanic will fix it. That is probably vertical 3. And if you want to understand how a motor car works the way it works, you will have to understand engineering. That is something like vertical 2. However, if you want to understand the principles of mechanics, principles of thermodynamics, principles of how a particular energy can be converted into another energy, that is basic sciences. So we would like to somehow see how to work with the techniques when we are confronted with a particular problem, right? In the group, there are people who are doing bachelor's degree in sciences or in computer science and in pharmacy or engineering or other branches like agriculture, for example, or they are doing master's degree, a master in pharmacy, and there are some postdocs and probably some junior scientists are there, and there are some established scientists who are there, and there are some people who are working in industry, and there are some people who are just freelance innovators. They just wanted to discover something big, and there are some people who are trying to just seek knowledge, and there are probably some people who are world leaders in this particular area. So different classes of people require different types of tools to play with, right? For example, if I need a car, I may use a Mercedes-Benz car or a Maruti or a Tata Nano or whatever car. So if a BSc student or an MSc student or a PhD or a postdoc student would like to use these tools, it is very, very important that they have to understand the principles behind that. And it is quite undesirable that people will get addicted to driving a Mercedes without really understanding any principles. Probably it's not even required. The mileage is wasted. So I feel that those who are in the academic arena has to learn the fundamentals. They have to be very good in vertical 1 and vertical 2, and then try to use as far as possible some of the open source tools that are available, or they have to develop these tools, which is very important. And developing these tools is an extremely important area. However, there are some people who are 100% involved in doing a computational work. And there are some others who are involved in 50% computation and they are doing the experiments. There are some others who are mostly doing experimental work and then they will be doing a smaller part of computation. So for the third category, where they are mostly doing experiments, they can be using a ready-made program, but however, they have to understand the principles behind that. But those people who are perfectly doing the computational work, it is extremely important when they are in the academic institutes to work on the development of these methods, understand these methods, and try to contribute to the development of these methods and why a program works the way it works. In contrast, if some of you feel that you need to have a computer and then you need to have a software and then get a training for 1 month or 2 months and you become computer-aided drug design people, my dear friends, I'm very sorry that it is not as straightforward as you think. Because one has to be very clear about what is the technology behind a particular work, a particular tool. It may be okay that you can run a car and then you can be a mechanic, you can fix, but will they be able to design better engines? You need vertical 2 and vertical 3. Engineers are required and scientists are required. So it may be very much possible that some of the mechanics think that they know automobile engineering more than an engineer or a basic scientist. Probably some others will also believe that he is extremely good and they can have their own innovative ways of making it. But unless somebody is very good in vertical 1 and vertical 2 and then try to understand that they have to be masters behind, they have to be masters in the principles behind these tools, it will be not a very advisable path. And I would say that it may be very detrimental to our education system. I don't want to mince any words in conveying the message to the people who are in the academic circles to be very seriously taking up the technologies that are involved in the computer-aided drug design. And our country with 1.3 billion people should, if they want to become leaders in the world, they have to have a robust manpower which will be aiming at developing our own software. With these few words, I will be happy to listen to Anamika, Nagamani, Himakshi and Lijo, who will take you to the journey of MPDS, which we have just started. Thank you very much, Giri, for giving me this opportunity. And then I will hand it over to the next people to take it over.
Good evening, everyone. So I hope you all are having a great time learning from experts in this training program of Drug Discovery Hackathon 2020. So I am Anamika Singh, a member of Dr. Shastri's group. Today, along with other group members, we will provide you with some background on MPDS and open source tool for computer-aided drug discovery. So the outline of the today's talk is like I will be talking about development of integrated drug discovery web portals, that is molecular property diagnostic suite MPDS. Another colleague will talk about MPDS modules, how they are, what is the schema of MPDS, and then Ms. Himakshi, she will talk about bioinformatics tools in MPDS and other tools, how to use them. And then Mr. Lijo will talk about chemical space and human informatics using MPDS. So the genesis of MPDS, molecular property diagnostic suite, the most common problem which is faced by synthetic chemists in understanding what molecule to synthesize and what not to synthesize, and therefore the knowledge of the properties we need in a molecule is crucial before synthesis. So we need to understand what are the properties which are required and what we need to synthesize. So if a molecule is there, if you give a molecule and if you are able to know its properties, then you can synthesize a molecule with desired property. So this is as the name suggests, molecular property diagnostic suite. Our idea is to have a portal where if you give a molecule, it will give you its properties. So it can be a drug-like property, biological activities, catalytic activity, fluorescence, etc. So this idea was conceived in 2004, and later the MPDS team started working in this in 2011. Though, and later the first version of our MPDS portal came in 2017, and there were many people involved, all from all over India. Several groups were involved, and this publication appeared as a cover page. And the MPDS is a series of disease specific web portals which are aimed to gather, find and discover all possible information on a particular disease from all possible resources, and along with that we add several CAD tools at one single platform. So the basic idea is to integrate and develop all open source together in a single platform along with the disease specific information. So here we have a web portal which provides a platform to integrate the chemoinformatics, bioinformatics, molecular modeling methods, that can be structure based or ligand based approach for computer aided drug design. They are integrated in one single platform. So for that platform we add chemical space, biological space, bioinformatics tools, chemoinformatics tools, and molecular modeling tools. So we develop these tools and also integrate the open source available tools. And this platform is disease specific, so it is which is created for the continual updation. It will be continuously updated, and there will be data related to literature, health, etc. So this is the home page of MPDS, and this is Molecular Property Diagnostic Source, which is a comprehensive indigenous end-to-end online tool for chemoinformatics, and we aim to assess the property of a given molecule and for the treatment of several diseases like tuberculosis, diabetes mellitus, and metabolic disorder. So this platform consists of several tools. You can see in the left panel, and this is the home page, and the executed jobs will be shown in the right panel. These two web portals are hosted for tuberculosis and for DM are hosted, and the links are shown below. Below, and we will be talking more about these tools later during the presentation. So how this MPDS disease specific portals are designed. So for that we actually have the Galaxy interface, which is designed. This is a user interface, and then we have at the Galaxy interface, and at the backend we for the chemical space and biological libraries we have the libraries, and which have the disease specific data as well as disease independent data. Tools are there, that is molecular modeling tools, chemoinformatics tools, bioinformatics tools. So what happens in this, the Galaxy in this one can add scripts or tools, programs in any language, written in any particular language, and it can be integrated, it can be hooked to the Galaxy portals and made available to the public as a tool. So for developing this particular MPDS web portal, we use the Galaxy platform. So why we use the Galaxy platform? Because it is a Galaxy platform provides a very flexible interface to add tools. So it has like four section. It has toolbox, job manager, model, and web interface. In the toolbox, it manages all computational tools. In job manager, it deals with the details of executing tools, and in web interface, it provides in Galaxy instant through a web browser, so it can be accessed worldwide globally. And then in the model, it provides an abstract interface for working with several types of data sets. So this is the flexibility of Galaxy portal, and that is why we have used this for development of MPDS web portals. And one of the most important thing about the Galaxy MPDS web portals are the integrated workflows. So workflows is like you can perform something one after other. So in this, if a CAD workflow is there, so all the steps of a CAD workflow can be performed one after other. So one can create a workflow and then come again and then execute the workflow with other data. So that is why these workflows are very important, and these workflows provides a very flexible way to run jobs in the MPDS web portal. And furthermore, one can add several tools to the MPDS Galaxy. So anybody can write a program or a script in any programming language, and then that can be for a particular given problem in CAD or the related area like text mining, data science, ML, which are working for the MPDS. And then we can add that to the MPDS Galaxy and make that available as a tool. So I will just show in a simple example of script and how it can be added to MPDS Galaxy. Suppose this is is a script to execute a GC output in a sequence, protein sequence. So this is a Perl script example, and this is an XML script example. So when you have a Perl script, this you can write anywhere in your system, and then we can integrate it to the MPDS Galaxy. And then this is a XML file where you write in the section here, it is written as tool example.pl, and we provide the format how the input and output is given. And then this tool this XML file is integrated to the Galaxy internally, and then you can, when the Galaxy portal is hosted, you can see the tool as a. So this is one of the example how any tool tool can be customized to an MPDS Galaxy. So and so one can write scripts, and then it can be hooked to the MPDS Galaxy. Similarly, we have developed several tools, these specific web portals. Two of them has been already published, which I talked about, like MPDS TB and MPDS TM, and several others are under the pipeline and are being developed by our team. And this can be accessed as mpds.neist.rest.in. And this is all about the introduction about the how this MPDS idea was conceived and how this has gone through till now. And further for the detailed information on how the MPDS tools are there and their modules are there, I will hand over this presentation to Dr. Nagamani. Sir, please take over.
Thank you, Aramika. And in my presentation, I would like to explain about what are the modules and the tools available in the MPDS in detail. Before I start my presentation, I would like to emphasize the call of our Honorable Prime Minister, how to become a strong international player in the area of computational drug discovery. For this, we essentially need a paradigm shift towards invent, innovate, discover in India rather than upping the West and use in India. So, as mentioned by our Prime Minister, this idea, before making this statement, this idea was conceived and being practiced in our group since 2011, which is known as this molecular property diagnostic suite. So here actually we have different type of modules, which is known as data library, data processing, data analysis, and recently we started adding advanced modules as well as we are working on disease mechanism. So essentially the available modules can be categorized into two different parts. One is disease dependent, another one is disease independent. If you see in the data library, we have the various section called target library, where we can get the information about the particular target. This will be vary for each and every different web portal. For example, in case of diabetes, we will get the information about the diabetes. In case of tuberculosis, we will get the target information about the tuberculosis. As well as the literature, we will get the information about the pathways and the available literatures, all those information about the particular diseases. But the gene library and compound library, these are all disease independent modules. So this is common. This is common means, for example, gene library, we have the library of all the human genes, as well as another one more very mega project under this MPDS is compound library, which we have collected nearly 130 million of compounds, and my another colleague will speak more about this one. And the data processing. So once we have the information about the particular target, as well as you have your own input, so how you will process? For that we have the another section where we can convert the file format. As most of the bioinformatics students knows, the file format will be in the PDB or SDF or MOL. So we have to convert in order to effectively process in different program. And the descriptor calculation, so we can calculate different type of descriptor for each and every molecule in the data processing module. And the main part is data analysis. So here actually we can do the different computational drug discovery calculations such as molecular docking, 2D QSAR, and recently we have added virtual screening, and we also have the drug repurposing, which we have collated the past server with one of our Russian collaborator. And once we perform the calculation, we can visualize the protein ligand complexes in the same platform. And recently we have added another module where we can do the sequence alignment, and we can also predict the active site in the data analysis modules. And advanced modules, and most of the advanced modules we are continuously working, and here as of now we have incorporated various machine learning algorithms where you can give your input and we can process in our tool and it will give you the output. And we also try to incorporate text mining, predictive computing, and multi-scale modeling. And another one more module is disease mechanism. We would like to understand the disease mechanism, and we are performing different type of protein-protein interaction analysis, and by means of protein-protein, we just trying to analyze what is the interrelation between the two different diseases. So this information also, this analysis also, near future we will incorporate in our MPDS suite of modules. And these are the how essentially that we have collated, how we integrate that this open source drug discovery modules in our MPDS, and how the cycle is working, how we are giving the input and how we are getting the output. So if anyone have their own input molecule from their local computer, they can access our MPDS homepage by giving the above mentioned that website. So there we can give the user input. User input is nothing but you can give the protein molecule as well as we can give the small molecules where you want to perform the calculation. So once you give the once you import this all this information, so that will be processed in our MPDS Galaxy server. In the back end, in our Galaxy server, we already have installed molecular docking, virtual screening, different type of modules. And we have written our indigenously script, which has been written in our own group, which will connect from this MPDS server with the open source modules. So when the user give the input, it will access the home page and it will submit it through our Galaxy server. Then calculation will be done in our local server. Then it can be accessed through our MPDS, this output page. So as I mentioned earlier, the input will be the protein molecule or small molecule, but once you give the calculation, then output essentially you can get the, in case of molecular docking, you will get the docking score, as well as you can download your protein ligand complexes also. In case of small molecule, in small molecule descriptor calculation, you can calculate the different type of descriptors by means of the Drulito, as well as we can calculate the ADME properties also. And this is actually the essential typical workflow, how everything is collected in a single platform. For example, let's say like I want to identify some small, what I can say, that small drug molecule against Mycobacterium tuberculosis. So you can access the MPDS TB homepage. So there you can get all the information about the particular MTB, and you can get the pathway. And in the MPDS target library, there you can get all the available protein targets for MTB. And in the gene library, in case if you have, apart from whatever we have deposited, if you have your own gene, those information also already we have deposited in our database that is available in the MPDS gene library. So from here, you can identify the target and you can process. On the other hand, we have the compound library, including FDA approved drugs. So there you can get the molecule which you want. And if you want to convert the molecular file format, for example, that if it is in mole file format, if you want to convert from mole to SDF, there you can do the file format conversion. And after that, you can calculate the drug likeness properties. And after that, you can filter the toxicity. And essentially that biological activity also you can, biological activity filter also we have, which will show whether the particular compounds have the antimycobacterial property or not. After that, with the identified the protein target, you can do the molecular docking, and with the by use of Autodoc Vina, then the result you can visualize in the same MPDS platform using the JMOL. And this is the overall the step-wise procedure available in the MPDS. And I thank my mentor, Dr. G.N. Sastry, to give me this opportunity. Now I hand over this session to Dr. Himakshi Sharma.
A very good evening, everyone. So here I am going to talk how MPDS can solve bioinformatics related problem. So if we see, MPDS is a package where all the bioinformatics related tools are integrated in a single platform. So this is the overall view of MPDS. Dr. Nagamani just now he has explained. And apart from chemoinformatics, CAD tools, visualization tools, we can also integrate various other tools like homology and similarity tools and protein functional analysis tools. Recently, we have added a multiple sequence and alignment tool in our COVID portal, as well as phylogenetic analysis tool. So, dear participants, here today I am going to demonstrate few of the computer-aided drug designing tools in our MPDS. So first I'm going to talk about molecular docking. So this is the overall representation of molecular docking. For that, we should get the data, for example, receptor and ligand, and receptor ligand, both the file should be same, that is PDB format. Before docking, you can also assign active site information by selecting, if you know the some active site residues, and before the docking process, you can also optimize your ligand, and then after performing the docking, you will get the complex file which you can analyze in the data analysis module, that is 9, and you will get the information about hydrogen bond, hydrophobic interactions, etc. So this is the steps of docking. So first you can optimize your ligand, And then you can also perform docking without optimization of ligand or with optimization of ligand. Then you can choose the docking with advanced feature, and you can also generate your conformation of your ligand to perform docking. So in this page, where you will see, if you go to our web portal, you will see the optimization option. In this page, you have to first select the ligand input file. So if your ligand input file is SDF, then you have to select the SDF file format. Then you should input the ligand, and you have to convert your ligand to PDB file format, because docking will only occur in PDB file format. After that, this is the doc version. So here you have to first upload your receptor file in the PDB file format, and as well as you have to select your ligand, which in the beginning you have converted in the PDB file format. That ligand you have to upload, and you have to select your residue, for example 309, and your chain ID you should give very carefully. If your protein contains chain A, then you should give chain A, and how many number of residues are there in your protein, and what residues this threonine contain. And then you can give any username doc, and you can execute. Then you will get your ligand and protein complex, and this is the PDB file format. So this page, it is showing you can also you can load the ligand, and you can generate many conformations as you like. Suppose, for example, here we have given 4, and you can execute it. You will get the 4 conformations of your ligand, which you can use for docking. Now, second tool is drug likeliness filter. Here we have incorporated many filters, for example, Lipinski rule, Weber's rule, ghost filters. So different drug-like filters have different descriptors. So before screening the molecules based on these filters, you need to do the descriptor calculation. So this page showing the descriptor calculation option. So you have to upload your given molecule, and you should execute. So you can see below here, molecular weight, molecular refractivity, polar surface area. These are the descriptors. So after calculating this descriptor, now you can select the molecule. You can assign the molecule, and then you will get this kind of things. So suppose for first molecule, it will show whether it will contain Lipinski rule or not. So it will, if it is there, Lipinski rule is following, so it will plus sign you will see. If it doesn't follow the ghost filter, then it will show minus. And if it doesn't follow the CMC filter, it will show minus. So I request Mr. Lijo to carry the further session.
Good evening, one and all. So I'm Lijo John, and I'm working as a research scholar in Dr. Shastri's group, and I will talk to you regarding the chemical space and cheminformatics, which is a great need for employing the various AI and ML and deep learning techniques in combination with the conventional modeling approaches, and this will be very much useful for chemical space as well as finding effective molecules for the various diseases. So let me start with the compound library, how we have created it and what are the unique features. I'll try to speak on these aspects. So as you can see, this compound library, we have created it by trying to pull the compounds. We have pulled the compounds from the various public domain chemical databases, and then we have tried to do some kind of data processing steps where we have removed some of the salt containing compounds as well as some of the other compounds which had some problems. So that was the initial part of the data processing, and then we have tried to classify them into portions. So these portions were are actually based on certain atom compositions as well as the position of these specific atoms, whether they are on the ring or whether they are off the ring. So based on some of the rules, we have tried to classify these chemical databases, and then we have obtained almost 97 portions. And then uh, for each of the database which was used to classify them into a portions, we have done the redundancy check so as to remove some of the duplicate molecules, and then we have obtained the non-redundant molecules. So once these non-redundant molecules are obtained, and you do this kind of step for all the databases, so this is like a loop you try to do for one database, and then you try to do for all the remaining databases, and then you merge the portions, and then perform another redundancy check, and then you also assign the bits. So the importance of bits here is to mention, or is to know these molecules, whatever we have taken, this comes from which all databases. So as I have told you that we have considered 37 open databases, so we have tried to generate bits for all these the non-redundant molecules, and then these bits were actually in the form, or in the this represents this belongs to which database, and the total number of molecules that have obtained at the final procedure was 122.124 million molecules. And now I will try to show you the 37 bits that we have obtained. 37 bits, I'll try to show you how the 37 bits was there obtained in the for each molecule. Before that, I also want to show you the Aadhaar card, and this is an card where you will get the all the information pertaining to a molecule, and all this information are containing the physicochemical properties of every molecule, and these physicochemical properties are can be used to assess their drug likeliness. And apart from all these physicochemical properties, every molecule here in this Aadhaar card is represented by a unique ID called MPDS ID. So you can see here that there is a ID mentioned here for a molecule that is atorvastatin. So then you have the first two digits of this ID. It represents the class. So class is nothing but a chemically well-defined group. The chemically well-defined group is in our definition we call them as class. And then we have the second number here that you see on the figure. So that is nothing but it represents the cluster. The cluster is a set of compounds where we have taken nearly 250,000 compounds. We have kept them in one cluster, and the remaining clusters are also arranged with the same order of the molecules. And then we have something called serial number. That serial number is nothing but it is just a number, and it keeps, as the molecules are keep on adding to a particular class and cluster, the serial number will be it will also increase. So this is the speciality of this Aadhaar card. It is a unique concept and completely Indian initiative. And apart from the properties which are being displayed on this card, we also have collected various other information, that is from which database the molecules were taken. So that is stored in the second page of the Aadhaar card. So what you see here, it is the first page, and then we also have loaded and compiled various other informations like spectral pharmacology and other toxicity information on the third, fourth, and fifth Aadhaar card. So all this information you will find in the MPDS Aadhaar card. Moving on to the further, yeah, as I have said that I will show you the bits here. So you can see this. This is one of the compound called bivaluridine, and it is there in the, according to us, it we have classified it in the first portion number 1, and then you see that we have represented it using ones and zeros, so on and off bits. So this there were, as I've said, we have taken the molecules from 37 databases. So and every digit here will represent, if there is for example, if the first digit is 1, that means that the molecule is present in one database, and then it is 0, so it means that it is not present in the second database. So like that, we have tried to put the information of all for all the molecules in this bit kind of representation. Yeah. Now I will show you something that how can you use the MPDS chemical space to search many compounds. So there is an option where you can import the data to the MPDS, and then that can be also, either you can paste it directly in the URL box, which is here. You can directly paste the molecule information. It can be a smile, or it can be a mole format. You can directly paste it here and you can execute the thing. The second option is also there, where you can draw the structure of the molecule. So we have the molecule editor there. You can directly draw the structure, and then it can be imported, and you can execute the search so as to get the molecule information. And there are various other forms of search. You can also search the MPDS compound library by using the database ID search. That is one form of search, where you can just put the MPDS ID, what I have already stated before, where you can put the MPDS ID of the database and search for that. And also you can perform the exact structure search, where the input can be a smile format, a mole, SDF, or a mol2 format. And also there is a possibility to do the substructure search, which again uses the same input as we have seen in the exact structure search. And then we have some search called molecular property based search, where you can search the molecules based on their molecular formula, molecular weight, as well as the other different properties. And then you have something called fingerprint based search, where you can search the cyclic molecules, acyclic molecules, and if it is acyclic molecules, how many rings are there, and also based on the number of heteroatom that is there in the ring. So you using all these different search aspects, there is a possibility of searching the chemical space in MPDS compound library. So here is this is like just a demonstration wise what I'm showing you here, where there is ID, database ID search option. You can directly put the ID here, and then you can execute the search. You will get the information, as well as you can also do the search, where you can also put something called substructure search, where you can put the substructure of a molecule and then give the substructure there as a input, and then you can execute, and then you will get all the substructure related search hits it will be obtained. So we also have something called molecular property based search, where you can put some of the properties, and then there is the possibility or something where you can use some of the conditions, where you can put and condition or condition, and then you can also insert more of the descriptors and perform the search. So this is molecular property based search, where the hits will be obtained in this kind of fashion, where you have the source ID, molecular formula, molecular weight, and all the related informations will be displayed in the search result. Then you have something called fingerprint based search, where you can do the search based, if it is a cyclic compound, you can select as a cyclic, or if it is acyclic, you can also select the option called acyclic there, and then perform. Then you can select whether the how many rings you want: 1 ring, 2 ring, 3 ring, and the possibility of different heteroaromatic information or homoaromatic. All these kind of descriptors are provided there in the search, and you can execute the search, and you will get the hits like this. So these hits are all are represented in the form of the Aadhaar card there. All the information of those molecules along with the search results will be displayed. There is also a possibility called you can actually input the scaffold of your interest, and then you can search by using that particular scaffold, and then the information can be obtained like this. This is also called the molecular cloud. And then we have something called library generator, where you don't want to search by any structure wise, but you want to see whether based on some of the composition of elements. Say for example, this is a very basic information, very basic example that we have put, that you please show the number of molecules that has 6 carbon and 6 hydrogen. Definitely you will get a lot of hits, but you can always try to restrain or restrict your search by using much complicated search queries also. So these are the various search options that is available in the MPDS compound library. So this is a hit, or this